So as you know, this is a joint organization between those two centers that are NIH funded. And one of our missions is to help promote the use of the software, the methodologies that we have developed. And also the idea is also to have new collaborations. We welcome new collaborations. So if you learn these things, if you like these things, and if you would like to apply them in your studies or in your research, we're here to interact with you. So this workshop is, you can see it as the beginning of a nice journey. It's not, when you live here, if you hate all these things, you don't revisit them. But if you really like, you can continue to work, and we will be here to interact with you if needed. Now today we have, we're actually switching gears. It is, all of you said, I came here to learn MD, most of you at least. So whatever you've learned, you learned, that's it. No more MD. Here we are gonna, well, there'll be a little bit, but mostly we are gonna talk about more advanced, more recent coarse-grained methodologies. And in particular, we're gonna talk about the elastic network models in the first lecture. Well, although today we will be, these are the three instructors, and tomorrow we're going to talk about druggability. And for those of you who like MD, we will revisit MD in the context of understanding how a given target is druggable. Okay? And also, we have been talking about the structure all the time, but actually it is always useful to combine sequence and structure information and have a better understanding of what has been conserved, what is co-evolving along the sequence, and then correlate it with the structure and dynamics. So this is this component that we are going to visit tomorrow as well. So we start with the lectures today. So I will give you a brief outline of the state of the art. And after the coffee break, Tim actually will go more into the details of the theory and applications. And then you're going to have this hands-on session with the help of Chakra and others. So here are actually two papers I wanted to briefly mention. One of them is, by the way, you already have a PDF file online. However, this is an updated version. I'm going to replace that one, but most of the, these references, a lot of material is already there. And for those of you who are interested, you don't need to take notes because it will be available to you. And for those who are interested, these are the basic, most important papers. One is a general background, where you can see Tim is one of the co-authors. And the other one is an original study which actually led to the development of ProD, which is the software that we would like to introduce here. So speaking about ProD, here is the, all right, I think that's better. So here is Ahmet, who is the father of ProD. He has not only, conceptually thought about building such a software that would be broadly used, but he also has been a leader in securing funds to support that. So there is, and Anindita, who just graduated, well, just is already a year ago. She also contributed a lot, especially to the new version of the ProD. Both Anindita and Ahmet were first co-authors in that. And so I would say that in the first place, Ahmet, and then lots of contributions from Anindita in the second version. And of course, you don't need the photos here. You've already seen them, Tim and Chakra. Ying was a former student who has been, his name is here, who has been very helpful in building the sequence component of ProD. Mao is will be serving as an assistant professor. He's also, his name is here. And then finally we have, as I mentioned, Indira, who will be helping this training with the drugability component. I think you will love that part. So this is just a motivation slide, just to give you an idea of what Prodi does. So we're going to explore Prodi, the software Prodi. And the goal is for you to be comfortable using it. Okay? So here's an example, and there are many features, but the way it started at this is the following. Suppose you have a query sequence. By the way, do you hear me? Well, okay. Suppose you have a query sequence, and this may be an entire protein or a segment. And you would like to know what type of structural data is available, or what would be the dynamics of that particular sequence, the corresponding structures. Okay? So you give as input, you enter the sequence of that particular sequence to Prodi. That's one type of input in FASTA format, for example. And what Prodi is going to do is to search the PDB. You all know what the PDB is, and you all know what FASTA format is. That's one way of entering the sequence. It searches the PDB to find all structures that contain that particular sequence. And not necessarily the identical sequence. You can define a certain sequence similarity threshold, identity threshold. So you can extract as many structures as you like if you keep it really broad. You know. And now you have an ensemble of structures. Okay? The typical applications would be, suppose you have HIV protease structure sequence. If you enter the HIV protease sequence there, it's going to retrieve from the PDB anything that contains the HIV protease or its orthologs, homologs. Okay? And this is useful, for example, for a given drug target, because those many structures in the case of HIV protease are going to be structures that contain a drug. And you will see slight structural variations between those different components, those different structures in the presence of different drugs. Or it could be another protein in the presence of a DNA, for those of you who are interested in the interactions with the transcription machinery. So you will have for that particular enzyme all the available structures, which you start to analyze. Now you can, here's an example, P38 kinase, which is a very important drug target. So you see here the superposition of those structures. In this case, although this is a large ensemble, they're really very narrowly distributed. So you see there. Now, using this ensemble, we do what is called a principal component analysis, and I'll explain that. Based on the principal component analysis, we identify what are the major structural variations. So you can think of some average coordinates and how much this ensemble of structures deviate from that average coordinate. You can think of it as a superposition of several modes of structural variations. And you want to extract the principal modes of structural variations. Okay? So when, for example, we analyze this ensemble, and we call it the experimental set, because these are all X-ray crystallographic or NMR models or resolved structures. So by principal component analysis, we're going to have here, if we focus on the most, first principal mode of variation, we can draw the arrows here, the violet arrows, to show what is the major structural change in the ensemble. That's one thing. Now we leave it aside. This is the experimental data. Now for your sequence, you know what the structure is and what the structural variation is. Okay? Then we build what is called an elastic network model. And that's also a very important component of Prodi, and that's how everything was initiated. This is a view of a network model. Does it look like a network? it does. It's a network, you can see. And for this network model, what we will see is you can calculate a unique set of accessible modes of motions. So for each structure, you have some what we call intrinsic dynamics. You have some motions that are intrinsically allowable. For example, my body is wired such that I can do these motions, but I can't do some others, okay? Depending on the way, if I approximate my body with a network model, I will know which type of flexibilities I have. And some of them are easier than others. So the network model gives you information on the different types of motions that are accessible, and that's what we would like to learn. And you can also obtain this. These are now the green arrows. And it turns out that in many cases, those most important, at least the first few, most important modes from the theoretical analysis and from experimental data analysis, they turn out to show good overlap. So we start to plot them against each other to see the correlation cosine between different modes. So that gives you really a full view, perspective of what has been observed, what is accessible, which may not have been observed, and that which may be exploited for further studies. So here is a simple conceptual example. Suppose an enzyme is doing this type of motion, okay? And you want to obstruct this type. You want to inhibit the enzyme. Inhibiting the enzyme would be actually inhibiting its dynamics. You don't want the enzyme to do the type of conformational change that it is undergoing to accomplish its function. So this is the enzyme. My arm is an enzyme, okay? And you want to obstruct this type of mobility. And you're going to now bind a molecule, your drug. Where would you bind it to obstruct the mobility? What would be your target point, target site? Is this very vague, the question? Again, so you want to block this type of mobility, and you want to accomplish that with the help of a molecule, okay, which will stick somewhere and block the movement. Where would you stick it? Yeah, you all know it already. This in the hinge region or somewhere near my elbow. Because if I have something that stabilizes this region, blocks this region, I can't move. If I attach it here, it can still move. But now I'm fixing the mobility of those two different subdomains. So what we identify in the elastic network models are those hinge regions. This is the hinge region. Hinge regions that are most susceptible to perturbations. So if you play with those hinge sites, and some of you may already know that, it's going to be consequential. And then when you look at many, many drugs that have been discovered and structurally resolved in the presence of the target proteins, you will see that many of them are indeed located at the hinge sites of the domain separations or say, a cleft between the two domains and things like that. Okay, so far so good. If we go with this speed, we will never finish. But that was a general introduction. Now we'll go a little bit faster. So one thing I want to mention here, this type of analysis for a given protein, how long do you think it takes to determine all the accessible movements, to generate movies of those motions? How long do you think it takes on real time?
6 months.
6 months? Okay. Any other guess? I'm sorry?
If you have the crystal structures, a few hours maybe?
A few hours, if we have the... Okay. So 6 months, a few hours. Well, for this one, it will be within seconds. Okay? That's the good part. You've seen how MD simulations are expansive. And there is a good reason for that, because they consider all the detailed atomic interactions. Now, what we're doing is very coarse-grained. But for those of you who would like to have a quick and first estimate of the conformational mobility, the motions, that's an excellent starting point. And for many cases, that's not only the starting point, that's also the ending point when you talk about large structures of thousands of residues, which you cannot handle by MD simulations. So this is extremely efficient. Which one? But it is approximate and needs to be refined, by complementing with MD. This is bad. So what we have been doing here in this study is for a given protein, in the conformational space, we are showing where the conformations are located, those points. Each of them represents a conformation that have been generated with MD. And the blue ones have been generated by, okay, let me see. These blue ones have been generated by A&M. I'm sorry, the red ones have been generated by A&M, and the blue ones are the experimental data. Now, you can see by A&M, which is the network model, really covers all the experimental structures, provides a nice coverage of what has been observed. But MD may easily deviate for the experimental system. So we have shown in many examples, actually, that A&M, the network model, provides an excellent way of sampling the conformational space, and we'll elaborate on that. So this is being very broadly used. We have some statistics. Now, there are yearly statistics, so we have some more than 130,000 of downloads, etc. This is of course, a new software, but it is being now increasingly broadly used. I'm sure you've seen the tutorials pages, right? So these are the two tutorial pages. The tutorial page for the ProD is over there. Here, let me see if I can go to that page. If I have access to internet here. Yes. So that is the ProD page. And in the menu here, you're going to see the ProD. The first part does the tasks that I just described, retrieves structures, analyze them. The evolve component will be doing the sequence analysis, comparing, whenever there is a critical site, which we identify, like the Hinge site, is it conserved? You can check that. We can compare this, the multiple sequence alignment. You can identify whether those positions are evolutionarily conserved. Or if there are coupled motions, you may want to see whether the corresponding residues are also co-evolving. When one of them changes, when you go from one species to another, the other one, does it also change in a correlated way? Sometimes these are called compensating mutations. And drug GUI is for what? This is what we call the druggability simulations, okay? So it is some kind of MD package where the solution contains many drug-like fragments, and we try to understand what are the sites on the surface of your target protein which are most likely to bind those fragments, and whether there are such sites in the first place. So this means that your target is druggable, okay? And NMWiz comes from normal mode wizard, is the visualization component. So you will learn to use all those 4 components, and you have the rest here. So let me go back. So briefly, actually, we have established this high-performance computing center, as I mentioned, which is called, actually, the short name is BIOS. The main focus is to understand the spatial organization and temporal evolution of neurosignaling systems events. So those interested in particular in neurobiological systems or drug discovery for membrane proteins involved in neurosignaling or other enzymes. So we are in the process of working on this type of projects. It's a collaboration between PIT, CMU, Carnegie Mellon, the Salk Institute, and of course the Pittsburgh Supercomputing Center here. This could have been your photo here, by the way, the one we shot yesterday. So, and the goal in the center is essentially to generate data for mesoscopic scale. What is mesoscopic scale? So meso is something intermediate. It's not fully atomic. It's not totally coarse-grained where you lose all the structure, space information. It's intermediate, okay, mesoscopic. So I don't know if you are able to see, but we have several different levels of studies. One of them is the molecular modeling, and then you have the cellular modeling. So you have the Monte Carlo simulations in a cellular environment, focusing mostly on the synapse. And Marcus is one of the experts in that area. And then, in addition to being an expert in MD, and we have also the tissue level, you know, and we're trying to bridge between those different scales. Now, at this time, we are only dealing with those, you know, these are the different scales, from nanometers for membrane proteins to micrometers or higher scales. And essentially, we are trying to expand this region by filling, you know, to go from molecular to cellular systems. And one of them is MD simulations that are being, you know, we use several different versions like accelerated MD, Brownian simulations, but also the coarse-grained models, elastic networks, which we will talk about. Okay. And the idea is each structure encodes a unique dynamics. So I'm sure you've seen this type of diagrams. As you go to longer time scales, you go from quantum chemical and molecular mechanics to coarse-grained computations. And you can also explore larger time scales. And these are examples of different events at different time scales. One thing I would like you to remember, if you know, everything else gets forgotten, is we want informational structure is the bridge to understand function. But if you look at the static structure alone, it doesn't provide much insights. You want to see how the structure moves, how the structure functions. So the bridge between the structure and function is dynamics, okay? By the way, please do ask questions. And again, to show you a flavor of what the system does. This is an AMPA receptor, a tetrameric membrane protein, very important for glutamatergic signaling, glutamate signaling. And this is, the structure is deposited in the PDB. We take the structure and then try to see what are the most accessible motions. So that static fixed structure is animated now. And this is one of the motions that is most accessible to the tetramer. Now you know how the tetramer functions. The good thing is that we can identify some regions, like here, where the interregion distance varies a lot. Okay? Now, in the structure, in the PDB structure, this distance is around, let's say, 25 angstrom, which is too far for them to come together. But we are predicting that there is this type of movement. They may come together. And then this may be tested. And indeed, our collaborators are doing it. They have already done that. They have substituted cysteines at those positions. And why cysteines? They form disulfide bridges. They can form crosslinks. And what happens if they form crosslinks? Exactly. You obstruct the mobility, right? And then the receptor, in the presence of cysteines and an agent that catalyzes the reaction to form the disulfide bridge, loses its functionality. It doesn't perform anymore. So this is experimentally proven. At least it shows that, okay, if it were only staying like that, there would never be cysteine disulfide rich formation between those residues. But now, because it's able to do this type of motion, this gives you some perhaps ideas about one way of interfering with the dynamics and thereby function. Okay. Okay, these are other examples of motions that you can easily predict. This is an entire virus. Is there anybody interested in virology here? A little bit, okay. So this is the entire virus actually containing not only the protein capsid, but the interior RNA, in this case, the genetic material. Now we take the structure and we try to, which is a fixed structure, we take it and we try to understand what are the most likely movements. So you can see which regions move a lot and what might be a mechanism for perhaps the viral capsid opening, releasing the genetic material. Or this is another example where you see how the molecule undergoes some machinery, okay, some conformational changes, mostly around the hinge site. Okay? So these are examples of predictions that we make using the network models. We have very pretty images. So we use network models for large structures. That's a ribbon diagram, but we map it into the network model. And then we can examine large systems, long-time processes well beyond what MD can explore. We can incorporate structural data at multiple levels of resolution. And we base everything in the theories of physics, polymer physics. Actually, it originated from polymer physics, from rubber elasticity theory. Okay? And talking about polymer physics, this is the guy who established the basic foundations, the theory of elasticity, which we have been borrowing and adapting to proteins. Paul Flory, the Nobel laureate in 1974. Okay. Now, in a nutshell, what we're doing is to take a structure, map it into a network representation, and then try to understand what are the most likely motions. So there is a movie that a former student of mine created. Let me see if I can show it. Where is it? All right, I think it is. So for example, if you take adenylate kinase, we focus on the backbone, okay, of the protein, and then on the alpha carbons. And the alpha carbons are connected by elastic springs. Now we have the network representation of the same molecule, okay, a new view. And then we focus on, we evaluate what are the most likely modes of motions, which we call normal mode 1, normal mode 2, and normal mode 3. Did it go too fast? I think you got the idea, right? Oh, it starts again, actually. Again, focus on the alpha carbon, backbone and alpha carbons, which are connected by elastic springs, provided that they are within a certain cutoff distance of interaction. A new network representation for the enzyme, which is subjected to normal mode analysis. And you can identify what are the most likely, most cooperative motions. Okay, mode 1, mode 2. So you know what types of flexibility, what type of conformational rearrangements it can undergo, and what would be the hotspots to target. So that, and a bit of the theory, because all this is not magic. There is a very simple, very elegant theory behind it. So this is our network model, okay? And then for the network model, we have a position vector defining the position of each residue. The position vector. And now we have the inter-residue distances shown here. And then we would like to understand what are the changes in inter-residue distances, the fluctuations, okay? So, and this is nothing else than the difference between the fluctuation in one position and fluctuation in the other position in a vectorial form, okay? So the idea is the essential idea is under equilibrium conditions, under physiological conditions, what are the naturally accessible modes of motions? You can call them fluctuations, but they are very cooperative also. Some of them are really collective. Fluctuations may be misleading. It's not like a small vibration. It is quite collective movements. So the idea is to decompose the spectrum of accessible dynamics into the contribution of several modes of motions and identify the most cooperative ones. Okay? So to do that, so we have already defined, this is called the fluctuation vector. And for a protein of n residues, you're going to have a 3n dimensional vector. Why 3n? Because we live in a three-dimensional space. We have data x1, y1, z1, x2, y2, z2, etc. for all the n residues. And then we're going to suppose you have a very, very simple model. And indeed, original theory due to Rouse. Rouse is very interesting. I'm always very impressed by him. He wrote one paper in 1956, and everybody knows him. That's it. He made it. So with one paper. That's not impossible if the paper is very creative. Of course, right now it seems very simple to us, but back in 1956, this was a big discovery. So what Rouse thought is, let me construct a B-dense spring model. Okay? Now, compared to the protein, in a B-dense spring model where you have a linear chain, what is the most important difference compared to a protein? In a protein, you would have other links, what we call the tertiary contacts. Here, this is like an elongated chain, okay? No tertiary contacts. Everything is just the chain connectivity. Okay, so he said, this is my system, and the overall energy is going to be a sum over all the harmonic potentials. Because each of the springs, this one, delta R1, 2 square, delta R2, 3 square, etc., all of them moving away from their equilibrium value and multiplied by a force constant. Typical, force spring constant equation, 1/2 kx squared, you are familiar with. So applied to this particular system. Now, in this case, you can rewrite this sum. Remember, we said that this fluctuation in this distance may be written as a difference between the fluctuation in the individual positions of the nodes. So you can rewrite it like that. And the idea is we have a uniform force constant. Just don't worry about the force constant. Everything is purely geometric, by the way. And then you can define, that's what he defined. Actually, he borrowed this from electrical engineering or signal theory, because Kirchhoff Matrix has been used in other matrix disciplines for defining the connectivity of a network. So what this matrix means is it gives information on who is connected to who. So you have residues from 1 to n, from 1 to n. Whenever you have a 1 or -1 in the off-diagonal term, they are connected. The other terms that are not seen here are zeros. So here we have only the first off-diagonal lines because everything is connected to its first neighbor. Are you with me? And anything else is not connected. So and the diagonal term is the negative sum of off-diagonal terms, okay, in each row. And it gives you a measure of the coordination number. What's the coordination number? For example, for this residue, the coordination number is 2. Because it has one link here and another one. There are 2 other nodes that are coordinating it. So the diagonal terms, for the terminal residues, it's 1. For the internal, it's 2. And the off-diagonal terms give you information on who is connected. Now, if you take this matrix, you multiply by delta R1, this one, the fluctuation in one of the residues, and you also pre-multiply, this is post-multiply, pre-multiply by the same column, row, fluctuation vector. Well, if you perform this calculation, simple matrix multiplication, I hope you are not scared about matrix operations. If you do that, what are you going to obtain? If you do this calculation, you're going to obtain this equation. Okay? That is, this sum over harmonic potentials is nothing else than so this operation that looks scary is nothing else than one way of calculating this total potential. Now we can generalize that. In the proteins, I don't have necessarily a linear chain, but I'm going to use a Kirchhoff matrix that has also off-diagonal terms. And then I'm going to do the same type of multiplication, but now have a more complete representation of the potential. So the total potential is simply found by this multiplication, which this is the compact notation. So in proteins, the Kirchhoff matrix will have off-diagonal terms also populated. And this is the definition of the Kirchhoff matrix. -1 in a given off-diagonal term if the two residues are within the cutoff distance of interaction. 0 otherwise. And the diagonal terms are found from the negative sum of off-diagonal terms in a given row or column because it's a symmetric matrix. Okay. Now we have this is all the theory. We're done. Practically, for each structure, you take the, you look at the coordinates, and you evaluate what the Kirchhoff matrix is. So it's very similar to something you may be familiar with, contact maps. Are you familiar with contact maps? Okay, some of you are not from molecular biological background. Contact map would be a map for n by n, where you're going to have peaks at positions, or dots at positions between when the residues make contacts in the tertiary structure. So it is very similar. For example, of course, this region is populated because anything along the sequence is interacting. Okay? That region is populated because it looks like there is a sequence somewhere and another sequence somewhere that make close contacts. What can it be? What? Exactly. Your name was Evangelia is really good. She's, I'm sure many of you have the answer, but at least she's also sharing. So yeah, these are, you know, think of two strings of residues that are making contacts. So they are forming a beta strand, a sheet actually, 2 beta strands. And now if they are perpendicular, one of them is increasing, the other is decreasing. What is it? I'm looking at her, but I want someone else. So you have residue numbers increasing in one of them and decreasing in the other, and they are making contacts. What could it be? No. Alpha helices would make contacts between every third, fourth neighbor. So it will be a contact between 3 and 7, 4 and 8, 5 and 8. So it is, they both increase. Now one is increasing, the other is decreasing here.
Anti-parallel basis.
Perfect, yes. She already told me here. Perfect. Okay, these maps are really useful to have a first idea of where the, you know, what types of contacts are valid for the structure. But more importantly, this could be used as a tool, as I will show, to evaluate mean square fluctuations. Now, this is your Kirchhoff matrix. I'm not going to go into the mathematical derivation of that. It will take a couple of hours, or at least 1 hour. But what I want to do is the following. In statistical mechanics, you know that you can evaluate any average property by calculating the property under different conditions and taking an average, right? So this is the property, the delta Ri, that delta Rj. How the fluctuation in residue I and the fluctuation in residue J are correlated, the correlation cosine, okay? So we want to find a measure of the coupling between two residues. When one of them moves like that, does the other one also move in the same direction? In which case, if these are normalized vectors, the correlation cosine will be 1, right? They move together. If they are anti-correlated, one of them moves like that and the other in the opposite direction, the correlation will be -1. Or if they are independent, one is moving like that and the other one is orthonormal, it's going to be 0, cosine of 90 degrees. So this type of, we have information on the correlations. Now, for each conformation that is accessible, and each conformation is weighted by its Boltzmann factor, and we integrate and divide by the partition function. Those of you from physics chemistry would know the expression. But the bottom line is that this reduces to a very simple equation. What is it? Well, it turns out that this cross correlation between residue I and j is nothing else than the ijth element of the inverse of the Kirchhoff matrix. So you have a Kirchhoff matrix, you invert it, you look at its ijth element, you multiply by a constant, doesn't matter, we want the relative values. So you just look at the ijth element, and this ijth element will give you information on how coupled residues I and j are. Isn't it magic? You just construct the Kirchhoff matrix, invert it. Of course, I'm not telling you that actually this matrix is not invertible. This matrix, because the way it is constructed, the rows are not independent. But there is a very easy trick to get around it. So and the software does. So you invert it and you obtain the mean square the cross correlations. And if I is equal to j, you have the mean square fluctuation, the diagonal term. Of diagonal terms will be the cross correlations. As simple as that. Okay. So now there are many, many applications. The application to hemoglobin was one of the relatively early applications. It's already 10 years old. And so what it does, we can obtain here this theoretical curve, the red one, showing what the mean square fluctuation of every single residue is in hemoglobin. Now, we know the structure. We just construct the Kirchhoff matrix, invert it, and plot the diagonal terms. So we obtain this profile, theoretically. Well, experimentally, they have determined the B factors. Do you know what the B factors are? For X-ray crystallography, you know, if you go to the PDB next to the X, Y, Z coordinates, you're going to have also a column saying what the B factor is. So the PDB doesn't only report the mean coordinates, X, Y, Z, but it also reports the fluctuation in those mean positions, which is the B factor. So if the predicted fluctuations are reasonable, they should correlate with what has been reported there. Under X-ray crystallography conditions, some of them move more than others. So that is, and this is the experiment. You look at the blue and red curve. To obtain this in MD simulations, I don't know, you need at least perhaps, in the best scenario, you need days, or if not weeks. This is done in 5 seconds. This is obtained. So this is the beauty power of it. And it shows that this very simple model can really predict fluctuations very accurately. One more thing, and this is the cross correlation now. It's the data are I dot data are j, I versus j. Okay. Now, and there are regions that shows strong signals. What do you see in this? This is called a correlation diagram. What do you see here in this correlation diagram? Can you say something? A unique feature? What? Symmetry. Some kind of symmetry. That's good. So what I see is this part of the diagram. Oops, I'm sorry. This part of the diagram seems to be very correlated, and this seems correlated, and this block and this block are less correlated. Okay. So this tetrameric structure, hemoglobin is tetrameric, has 4 subunits: alpha 1, beta 1, alpha 2, beta 2. And actually, alpha 1 and beta 1 are very coupled to each other. And so are alpha 2 and beta 2. And they are, you know, alpha 2, beta 2. And they are decoupled from each other. And many people have been calling the hemoglobin a dimer of 2 dimers. Now we understand why, actually, and which dimers are coupled. these move very coherently, and these two, and they are decoupled from the other two. You see the type of, so we understand now, if you look at this structure, could you tell which one is coupled or decoupled? Very hard. But when we do this type of analysis, now we identify which pairs are moving together, which pairs are not. Okay? Which pair, dimeric portion. Questions?
Excuse me.
Sure.
I definitely like this plot, but I have to be very honest, I got lost.
I'm sorry?
I got lost already with this beautiful plot.
Yeah, this is very,
Symmetry is beautiful, but as some reason you color differently. Would you mind just elaborate a little bit more?
Sure.
So when we look at this, how we interpret alpha 1, beta 1, and can we get some
Sure.
information of the ligand?
and first of all, there is something important missing here. What's the color code? So these are, the violet is the strongest signal, followed by the cyan, and then followed by the brown, okay? And then white is like no signal, and orange would be even anti-correlation, negative correlation, okay? So everything along the diagonal is very strongly correlated. What does it mean? Whereas I moves, I plus 1 is also moving. You know, this is the connectivity. Everything, you know, the first neighbors, they are moving together, okay? And even there is this strong correlation that spreads to a larger sequence distance around the diagonal, okay? But we can already see some 4 blocks that seem to be moving more concertedly, you see? And this is actually the alpha 1 subunit. You have 4 subunits, alpha 1, alpha 2, beta 1, beta 2, okay? This is already the alpha 1 subunit that seems to be moving very concertedly. And then it shows some correlation with beta 1, okay? It is totally anti-correlated with alpha 2, and some correlation with beta 2. So that is how you see this map. Now, if I look from a really distance, what I see is there is a block here and a block here, right? This region seems to be more correlated, and this region more correlated. So alpha 1 and beta 1 are more correlated within themselves, and alpha 2 and beta 2. And they are anti-correlated with each other because these cross terms are 0 or very weak correlations. Okay?
In other words, actually, the information behind this table indicating which residue they're interacting. Right? So all the information there.
Yeah, for example, this tells me the boundaries of regions that move concertedly. So I have a block here, another block here. They move, but they move coherently, more or less, each of them, each of the subunits. But some subunits are more correlated than others with each other, the cross correlations.
Can we tell the ligand and receptor information?
No, there is no ligand receptor so far. This is just the analysis of the hemoglobin itself. But what happens is that usually there are weak points. You know, the crossover between different concerted regions will be the weak points we want to interfere with, in principle. I think I'm going to go faster a little bit. So you have seen this type of images, right? For example, when we take a structure from the PDB, they report the coordinates of this green molecule, but actually the crystal structure looks like that. In the crystal, you have multiple replica, you know, of the same molecule surrounding your resolved one. And depending on the crystallization condition, you may have different contacts, you may have different geometries, etc. This is the same molecule under 2 different crystallization conditions. And you can see that sometimes you may have this type of crystal contacts between neighboring molecules. And remember, I said in the crystal structure, you have also information on the B factors, how much each residue moves. Well, the thing is that under physiological conditions, there are no such contacts. So these are artifacts of the crystal packing. And the good news is that those artifacts, they don't usually affect much the coordinates. So the crystal structure is a good representation of the native structure under physiological conditions. We can take that. But when it comes to interpreting the fluctuations, of course, you would expect those regions to be more severely suppressed, right, because of intermolecular contacts. Are you with me? So when we compare with the theory, actually, what happened with for this molecule, we saw that the theory predicts large fluctuations at those positions, although the crystal contacts, the crystal B factors are very small. You know, this is the size of the fluctuation at those positions. And then we recalculated by building the environment, and it is so easy. You can, instead of 1 second, you can do it in 15 seconds. And then we repeat it, and then the theory reproduced the experimental data. So the theory became as wrong as the experimental data then by putting the same artifact. So that shows, that gives us also further confidence that we are predicting not only, we are reproducing experimental data, but what we are also calculating may be even better because it's devoid of those crystal contacts. All right. Normal modes. That is some of you who from physics, even high school physics, you may know that a given, each system has a distribution of vibrational modes. So if you have a spring, you may have a very cooperative motion, or you may have something that divides it into 2 parts or multiple parts, high frequency. So modes are different frequency motions that the structure can have access to under equilibrium conditions. So the idea is that when you have a certain cross-correlation, it is actually a summation of multiple modes. K index here refers to modes. Okay? And the idea always is to decompose the observed covariance, or the one predicted, into the components, into the different components, and extract the major contribution. Because otherwise it's a mess. You have multiple, it becomes very noisy. But we would like to extract what are the most cooperative events. And of course, there are evolutionary arguments saying that those most cooperative events ought to be functional. That's why this structure has been selected to do its function. And the structure should be favoring those dynamics that enable function. Okay? So we do this type of, and remember, this was found from the inverse of the Kirchhoff matrix. So the idea is then to decompose the matrix. We do an eigenvalue decomposition. Each eigenvector represents a mode, and then eigenvalue represents its scales with the frequency of the modes. So these are eigenvectors, eigenvalues. I'm not going to go into the details. But the idea is each structure, we can generate for each structure several different movements corresponding to different modes of motions. But usually the first 1, 2, 3 are the most important ones. So when we do such things, here is how the different modes look like. This is the first mode for HIB protease, actually. It looks really quite smooth. It is very cooperative. And here's how the highest frequency mode looks like. You're going to have only peaks at certain positions, which undergo, very sharp vibrational movements, very localized movements. So the high frequency modes, which are usually less interesting, will give you peaks. But these are regions that are really subject to very high energy on a local scale, and they cannot sustain any perturbation. So in fact, those residues, we have shown that they ought to be conserved because they serve like the nucleus of the protein. They are very highly constrained, packed. And this gives you a profile about, which region is doing what. So for example, this region is moving a lot. This is indeed the recognition loop of the HIV protease, which has 2 flaps. This is the hinge region between the 2 flaps. Or the active site is a minimum. This is a general rule. The active site will appear as a minimum. So it is like the cleft around which you have the domain movements. If the active site is very sloppy, it's not going to do its function. It needs to be very precise, precisely packed. So this is a very highly constrained region. And in the collective motions, what happens, it's like an anchor around which all other things are moving. Okay? This is a general design principle. And what we are essentially doing is we are near this energy minimum, and we are trying to explore what is happening in this neighborhood. So a summary. From the structure, you map into a network, you construct the Kirchhoff matrix, you calculate the covariance, etc., and you decompose it into the contribution of different modes. And this is like mapping the structure into obtaining a new map of the behavior of the structure in different modes. And you can do the same thing like that for the cross correlations too. Okay, this is just. I think I will. I'll skip that part. The anisotropic network model is an elastic network model that has been very broadly used and forms the basis of PRODI. So that gives you some kind of visualization. In mode 1, this molecule is going to do this type of movement. You know, the two domains move with respect to each other. In mode 2, they undergo some kind of counter movement. Well, in a higher mode, there is only a single residue movement. And if you want to plot the corresponding energies for each mode, these are fluctuations. These are like harmonic events. This is very smooth. It just happens very slowly. So the curvature is small, the force constant is small, the frequency is small. This one is very sharp. It is just very localized. So this is the representation of the energy variation as you move away. So you can move only a small distance, and it encounters very big resistance. All right. I think these allosteric changes so there are a few applications that I was considering to do, but I will leave it to the application session. So we will skip these things. And then I already mentioned this ensemble of structures. Ubiquitin has been always a prime example. What is ubiquitin? What does ubiquitin do? Sometimes they call it a kiss of death, because it binds an enzyme protein and then serves as a tag for directing it to the biodegradation machinery. So it's for killing the enzymes, for biodegrading them as it binds. And to bind it also, each time it binds a given protein, it adapts, you know, it changes its structure. And this shows the conformational variability. This part is really bad. So as I mentioned, we analyze ensembles of structures or a given protein in the presence of many different inhibitors. And from that, we can also calculate what are the types of motions that different machines undergo, alternative motions. And this gives us, this type of information gives us insights about molecular methods to control the dynamics. So, you know, when you do this type of analysis, usually we focus at interfaces. One of you was interested in interfaces, I think. You focus at interfaces because that is where there is a redistribution of interactions taking place. And the interfaces will give us, then we can identify which conformational switches take place. Here is an example for the same machine. You know, this interface, when it moves, the reds are negatively charged, blues are positively charged, and they are forming salt bridges. As the molecule undergoes a conformational change, the negative and positively charged residue, they switch partners. Okay? So the new conformation may be then stabilized because it's a conformational switch in a sense, because they change partners. And these are real. You know, people have been observing that such changes take place. This is just an evidence for that particular one, where you have a change in the neighborhood, in the partners, you can go you can go to this paper where, and then now if you are mutating a given residue at that position, you can you really interfere with this type of salt bridge switches, and then block, for example, this type of mobility. And certain mutants, this particular mutant is one of them, which would lock it in one position and prevent the movement. So when it locks it in that certain position, it will also interfere with the cycle, you know, a functional cycle this molecule is doing. So there are this type of arguments. This is, I'm a little bit worried about the timing, but we're doing well, not bad. If you have questions, please ask. No questions? So that is what we like to do. For a given structure, again, this is the same structure, we have an ensemble of modes of motions, correct? Okay, we have an ensemble, and these are the modes 1 to 25, the most probable 25 modes. And then for this structure, we know that there are two experimental structures. One is, and we were talking about, in this case, about a chaperone, molecular chaperone, actually, bacterial. It has a what is called a relaxed and tense form, R and T forms. And it undergoes a transition between them when it's functioning, okay? The chaperone inhibits folding of other proteins, and it will do so by undergoing a transition between R and T forms. This is an ATP-regulated machine, etc. Now, there is a structural difference between the R and T forms, and what we want to see is whether this difference in the structure is something the molecule intrinsically wants to do. How do we test that? Well, we evaluate those normal modes of motions, right? If a given mode of motion is such that it would provide the passage from R to T, then what the molecule is doing is just recruiting that particular mode to accomplish its function. Okay? And when we compare with individual, this one, by the way, this is a very good example I wanted to, not always, we don't get so lucky always, but that one, in this case, the first mode, which we think is the most cooperative motion for that subunit, you see this is one form. This is the, which one? This is the relax. One is the relaxed, the other is the tense. You would think that this is the relaxed, this is the tense, right? Because it's more compact. So during this transition, it undergoes a conformational change. So we evaluate what is experimentally observed. We have a 3-dimensional vector for the structural change experimentally observed. And we compare this 3-dimensional vector with our individual modes, which are each also 3-dimensional vectors. We look at the correlation cosine in between. And in this case, you can see that the first mode already provides a correlation cosine of 0.8, which means that the molecule is simply moving along its most easily accessible, most cooperative, intrinsic mode of motion to move from R to T. Or it has this path already built in it. There's a pre-existing path. Of course, this movement is triggered by ATP binding. Yes.
So I'm just curious, for this type of analysis, is it always true the first mode?
Not always. No, not necessarily. In the review that we wrote, you can see many examples. It is very interesting, but usually the second mode correlates more frequently. But one of the first, second, or third usually provide already make a substantial contribution to the passage. Now the idea is the following, and this is an ATP regulated machine. So this movement is actually triggered by ATP binding. So ATP binding is providing the energy to move a little bit from one direction to another and stabilizing a new conformation. But the idea is that the ATP binding alone would not do that if the molecule was not already prepared, was not already predisposed. So intrinsically, the molecule has this ability to move from one state to another, and the ATP is just triggering this pre-existing ability. And this is the same type of logic when we design drugs. molecules have certain tendency to move in certain directions, and you would like to design drugs to interfere with those particular movements. But those drugs that are pushing the molecule in a conformational change that is not readily accessible, they are not going to be effective. You see what I mean? All right. I will just skip this. We already spoke about that, the principal component analysis. So when you have an ensemble of structures, and this case is HIV reverse transcriptase, in the presence of different inhibitors, in the presence of DNA, RNA in the yellow, or in the apoform, in the absence of any ligand, we superimpose them. We calculate for the ensemble what is the covariance matrix for all the covariance is for every single residue. We evaluate the mean position in the ensemble and how much the mean position deviates for how much for each conformation the position of that residue deviates from its mean position. And then we take the cross correlations between everything. We generate an ensemble, a covariance matrix. Well, the covariance matrix is the inverse of what we spoke, like the Kirchhoff matrix. So you can either eigenvalue decompose the covariance or the connectivity matrix. You get the set of principal motions. And this is from experiments. So these are only experimental observations. You build the covariance based on the experimental observations. And you extract from here the principal mode of motion, which is this one. This part is called the finger. This is called the thumb. Thumb and finger tend to move with respect to each other. And this is where the DNA actually lies. So they process the DNA, the HIV reverse transcriptase. RNA, actually. What's happening? OK. So we can do this type of calculations for many different proteins, compare experiments and theory. And you will do many applications of it. So in principle, again, for the same molecule, we have this type of mobility, as you can see from the superposition of three structures. And when when we analyze the structure itself, the structure, and we do our A&M analysis, network analysis, this is also mode 2, by the way, A&M mode 2, which gives the same type of motion. And the ensemble of structures that have been resolved for HIV-RT, at least when this study was done, they all actually lie along this mode. So the structures are different. They differ in the extent of, opening of this region between the thumb and fingers. So there is not like two discrete states, but a distribution, as you can see here. And we can superimpose those predicted by A&M or those predicted by PC1 with a correlation of 0.99. So that's a good example, again, showing how these things correlate. This is my last slide, actually, where we are. Again, I will repeat the same concept. That's a very important message. Now, you compare the same protein, two different structures, OK? One is orange, the other is blue. And these are stabilized in the presence of two different inhibitors. So because one of the inhibitors favors a slightly more closed conformation, the other inhibitor favors a slightly more open. But then if you analyze this structure, it has indeed this intrinsic mobility. And it is going to accommodate the binding of the inhibitor, optimize its interaction because of this intrinsic mobility. Now, suppose you just look at the crystal structure and you see it stays like that. If you were looking at the crystal structure, then you would argue that, oh, no, it's impossible to bind something here. But once you know that actually it has this intrinsic mobility, you can generate an ensemble of alternative conformers and do your docking studies. And you will see that, indeed, it has a very nice binding pose for your hit target, for example, drug, for example. So that is why it is really important to have a better understanding of the conformational variability because that is one of the major problems in computer-aided drug discovery. If your target is fixed, if you don't take account of the conformational flexibility of your target, you're going to miss lots of accessible binding poses. Okay? This is a review summary, and which should be clearer after all this discussion. And I'm just, I don't know, I didn't know that. No, this acknowledgement doesn't apply here. But the Pittsburgh nice view applies. All right. So if you would like to ask a few questions, we have some time, I think. Yes.
I'd like to ask something.
Sure.
Sounds like a little bit of backward things. So definitely.
Backward is always good.
So definitely, this is a very efficient way to sample the conformation.
Absolutely.
It's always a challenging topic,
Absolutely.
no matter for small molecule or big protein. So and also one subtopic, even in MD field, we call it a replica exchange. Basically, we're starting from different starting point.
That's
then
true.
try to see whether it's converged at a certain point. But let's say in your way, you can quickly generate a lot of representative conformation.
Absolutely.
It might be totally different. But I just wonder whether some studies, starting your conformation and do some MD and try to see whether it converges at a certain point. I'm just kind of crazy and try to link.
It's
your
not
studies
crazy
with
at all. And what you said is actually what's being done.
I'm
So-
out of date.
You see, your intuition is totally correct. And, I didn't mention, but in the one of the first studies where we have combined MD and this type of predictions was a paper we published together with Klaus, actually, in 2008, I think. So the idea was the following. Now that we know that we have this type of domain movements accessible, but in MD simulations, you will never observe those. You will observe small fluctuations. But when we do our domain motions, these are also sometimes being not so realistic because some atomic interactions are being violated. So we want to combine them. Then what we did was to use those A&M modes as a guidance you may know about targeted MD, steered MD. So we use these modes that are predicted by the elastic network as targets to steer the simulations. So what we are observing is that a motion along these, the molecule is moving, undergoing those large motions, but at the same time, it is undergoing the right atomic interactions. Now, when we did this original study with Klaus in 2008, we selected certain modes. We said, OK, it's going to move along mode 1, 2, 3. But there is an arbitrariness here. And you have a whole pool of different modes with different frequencies. So more recently, last year, I think, we published another paper where each mode is being selected randomly, like a Monte Carlo. You have a pool of different modes. You just select one of them, and you let the molecule move a little bit. But when you deform the molecule along this mode by a very short targeted MD. Then you do just relaxed MD. And so there's a whole protocol. Then you select another mode randomly, not randomly, but in proportion to their natural frequencies of occurrence. So this combination of moving along the normal modes and then relaxation using the MD simulation, it is, I think, a very powerful tool, which is now being implemented. Chakra may give more information, which is being implemented in ProD. So we would like everybody to be able to use this type of methodology. But this is very new, It's, so what you said is not background. It's very forward-looking. And it is actually what is being done.
I'm definitely very interested in this technique.
Sure. So we can talk more.
Sure.
There was another question in the back.
I have a few quick questions.
A few, okay, go ahead.
I'm curious about your network of springs. The springs are different lengths. Are they given the same spring constant and the same initial total energy?
That's true. So there are, it's like a, hardcore potential. If, for example, two things are within a close proximity, they interact. Otherwise, they don't. So we took, in the network model, in the A&M, let's say, you take the cutoff distance of 10 angstrom. So anything within 10 angstrom, we connect by a spring. So we don't distinguish whether it is 5, 6, 7, 8, 9. So it's connected. That's all. And we assign an equal force constant to everything. It's so simple. All these things are approximations, but we've played with these things, tried to distinguish between different pairs, and it doesn't really provide a big improvement.
Okay, with the underlying spring model, so you get the modes. With the physical component analysis, you're able to get the modes. How do the wave numbers, how are the wave numbers?
Wave numbers, you mean the frequencies?
Like the frequencies,
Yeah. The frequencies are directly obtained from the eigenvalues simultaneously. When you get the modes, for each mode, you also obtain its corresponding weight, statistical weight, the frequency of occurrence. So it is the frequency.
Okay, I was wondering, so the modes are good, like, do you get,
It's
are you able to compare those to experiment with the spring model, or do you really have to go to something like MD or?
No. We don't compare with MD because the goal is actually, this has been done. Many people have already compared with MD. The goal is to go beyond MD. So there is no point comparing with MD if you are dealing with a system where you are observing cooperative motions. These things won't be observed by MD. But as a validation, verification, we did this type of comparisons. But the major utility is to apply these approximate methods to systems, large systems, and long times where you cannot really explore by MD. If it's a small system, and you should use, of course, as detailed as possible. MD is wonderful. But there are larger scale systems, more cooperative events, like where there is no way you can use MD. And although these are approximate, they give lots of useful insights.
Thank you. My final quick question is, do you have to account for rotational or translational motion that bleeds in from the numerics of it? And if so, what do you use to?
The theory, by definition, it's always calculating the internal modes. So for example, when we do the A&M, there are 6, if you are familiar, there are 6 zero eigenvalues, which refer to the 3 rigid body rotation and 3 translational degrees of freedom. So then we start the real movements, internal movements. So we focus on internal only, not the rigid body.
Very quick one. So the analysis will quickly generate so many number of normal modes. I just wonder, is there any quick way we can, let's say, focus on first 3 or first 5, which cover, let's say, 80%
Good.
of the motion?
That's a good point. For each mode, we have a statistical weight. So you can evaluate, for example, the first mode may be accounting for, let's say, 20% of all accessible movements. Usually for 10% of the modes will be more than enough, usually. A small subset, depending on the size of the molecule, let's say a typical enzyme of 300 residues, if you take the top 20 modes, that's more than enough, usually. It will so this is already a representative set, and the remaining will be, but in this Monte Carlo method, you can pick up any mode you like in proportion to their natural occurrence, so you don't have to neglect. And naturally, it's going to select from this top 20 because of their overwhelming, Yes? She's going to ask a very tough question, I know.
No, I was just thinking like transmembrane proteins versus ones that are in the cytosol. So for the transmembrane proteins, there is all the membrane that is not in the
This is a wonderful question which Tim is going to answer. You are ahead of us. You are not. She's done with this lecture. She's asking questions about the next lecture. So Tim will answer that. Okay. And there are good answers for that.
So the network model you have remind me a lot in NMR calculation.
Yes.
Can you comment on like link to the NMR function?
Well, actually, I understand perhaps where you are coming from. In the case of NMR, you have a bunch of distance constraints. And using those distance constraints from NOEs, you can construct the structure. That is, Here also, actually, we don't, you know, the ingredient, the major ingredient is this connectivity matrix, right? We don't need the structure. If we know only who is interacting with whom, a list of contacts, we can do the calculations. So in this respect, there is a similarity because everything is based on contacting pairs. The other issue is in NMR models, you have an ensemble that is being resolved because those distance constraints, they are not sufficient to fully determine the system. The system is not fully defined by that set. So to comply with it, it's like an optimization problem where you calculate a set of models. But this set of models that NMR spectroscopists are depositing are actually very valuable because they also give you about information about the type of structural variations that are actually compatible with their experimental data. And we've done this type of comparison, comparing the NMR models versus what we would predict as structural variations. There is a very close similarity.
So do you always define the beads on the residue, or
you can do anything you like. You can do anything you like. The default, in Prodi would be alpha carbons. But you can even, you can define some blocks. In virus dynamics, there are studies in the literature where a given virus, which is composed of, I don't remember, some 400 plus monomers, they put just one bit for each monomer. So thinking that each monomer is a, structural entity. This is a very coarse-grained approach. But even that yielded some valuable data and was published in the Journal of Molecular Biology. So you can define your structural block the way you like. And you can go even in the other direction. You can put a node to every single atom. But I would not go in that direction. That's killing the purpose, right? We want to go to higher scales.
it's always like a spherical?
Well, it's a node. It has no shape. It is an interaction center. Think of it, yeah. Yes?
I just had a quick question about cross-correlations. I know you mentioned it, but I think I missed it. So positively correlated is like motions in the same direction? And
Exactly.
correlated is like opposite direction?
Yeah, two things are positively correlated. If you and I, we walk together, and then we turn together, and then we are positively correlated. Now, if you are moving and I'm totally, somewhere else, there is no correlation. Because these are also averages. Then you take how often this residue goes in this direction and how often the other residue goes in the other direction. You take an average. And on the average, if you don't see, the correlation cosine is 0. So it is uncorrelated. Anti-correlated would be, actually, it's a strong coupling as well, anti-correlated. And it is indeed, whenever one residue moves like that, the other moves in the opposite direction. So they are correlated, but the sense of motion, the direction is the same, but the sense is opposite. Right. Yes?
I'm doing empty simulations, and for example, I want to compare the results of my simulation with the elastic network model.
Mm-hmm.
And it will give me the Hinge regions and
The elastic network model? Yeah.
and the most cooperative modes.
Yes.
Should I think that the Hinge regions, for example, should be mostly represent the ones in my empty simulations for a guide maybe or is there a constraint, for example, for the big structures? Is it also applicable to compare the two results?
Now, if you want to compare MD results and A&M results, there is a very simple way, and actually Prodi does that, by the way. Your MD trajectory, you can do what is called an essential dynamics analysis. Maybe you're familiar with. But the thing is, when you generate an MD simulation trajectory, you can also build a covariance matrix from the trajectory. You know the mean positions, how much each residue moves away from the mean position, take all the averages. So you can build the covariance matrix, and from the covariance matrix, you can obtain the principal modes of motions observed in MD. So those principal modes of motions may be compared with the elastic network model principal modes of motions. Now, normally, I would not expect to see a strong correlation unless in MD also you are starting to view those large domain movements. Maybe you, by this type of decomposition, you may discern some more cooperative events that may correlate with ANM. So we do have in ProD, I don't know if you are going to talk about that, but in ProD there is an option to do this type of trajectory analysis and extract from MD the most probable movements, modes of motions, and then this may be compared, if you like, with ANM modes. Okay.
One more question.
Yes.
Is it when you do the analysis of the normal modes, or like you produce that information, can you have a ligand in the protein versus one protein that does not have that ligand in there, and observe if there is a difference in the movement?
Yeah, the question is, if you have two structures, one with the ligand, the other one without the ligand, do you observe the difference? The answer is no. The answer is, think about it. When we take the network model, we have, let's say, 300 nodes in the absence of the ligand. With the ligand, you have 301 nodes, because the ligand itself will be perhaps one or two more nodes. So when we calculate the most cooperative movements, adding one node or subtracting it, it won't change the overall behavior. What we do instead is we do in the absence of the ligand usually, and try to see, what are the most accessible movements. And then we compare with those in the presence of ligand, and we see that one structural deformation that is predicted in the absence of the ligand, there are such papers, is actually what happens when binding the ligand. I can give you a reference where this is the whole. So the movements in the unbound state are those observed upon ligand binding. This is the title of a paper we wrote in 2005 into PNAS. I can give you the reference, yeah. All right, I think if there are no more difficult questions, I can take easy ones. But if there are no more questions, it's a good time to give a coffee break, or what is the plan? Right? Coffee break. And you are welcome to, of course, ask more questions in the meantime. And I'll be around all day, and we can interact as much as you like.